So in this video we're going to talk about how muscle fibers contract. And we need to talk about the microanatomy of skeletal muscle cells before we can really talk about their contraction mechanism. And we know that skeletal muscle fibers are long cylindrical cells that contain multiple nuclei. Because these cells can be up to feet in length, we have special terms to describe some of the components of these cells. Like the sarcolemma is the muscle fiber plasma membrane, the sarcoplasm is the muscle fiber cytoplasm, and skeletal muscles actually have a lot of glycosomes, and these are involved with glycogen storage. That way these cells can have a source of energy during peak activity. And myoglobin is used for excess O2 storage. That way skeletal muscles that rely heavily on oxygen can continue to produce contraction force even during peak activity. Now, skeletal muscles have modified organelles, like myofibrils, or these long protein cylinders that fill most of the muscle cell cytoplasm. And sarcoplasmic reticulum is the modified endoplasmic reticulum of muscle cells, whose function is to store calcium. And then T-tubules are essentially tubes of sarcolemma, or muscle fiber plasma membrane, that traverse deep into the cell, and their purpose is to basically transmit electrical impulses within the cell. Now, myofibrils are these densely packed rod-like elements of protein, and essentially each muscle cell can contain thousands of these, and they account for about 80% of the muscle cell volume. Now, myofibrils are the proteins that give these muscle cells their striation appearance, as well as containing what's called a sarcomere, which is the functional unit of muscle contraction. Now, sarcomeres are made of myofilaments. We have thick and thin myofilaments, and we'll talk about the molecular composition of these myofilaments as well. Now, this is showing the muscle cell, or fiber. Remember, the specialized plasma membrane of the muscle cell is called the sarcolemma, and deep within this you're going to find all of these protein cylinders. They're called the myofibrils. Now, myofibrils are made of repeating segments of sarcomeres. So a sarcomere would be about from here to here, repeating across. And these sarcomeres are made of thick and thin myofilaments, which we'll talk about the anatomy of here pretty soon. Now, striations are the stripes that are formed by repeating dark and light bands along the length of these myofibrils. So we see that if this is one skeletal muscle cell that contains thousands of myofibrils, the repeating segments of dark and light areas are basically what make the striations on this muscle cell, and it's essentially all the sarcomeres that are lined up and repeating in a way that allows these muscle cells to produce a tremendous amount of contraction force. Now, the sarcomere is the smallest contractile unit or functional unit of a muscle fiber or cell. It contains A bands with half of an I band at each end, and these are actually found between what are called Z discs. Now, individual sarcomeres align end-to-end along the myofibril like boxcars of a chain. That way, when one sarcomere contracts, it's going to pull on its surrounding sarcomeres. And in reality, really, it's just sarcomeres all along the length of this myofibril that contract around the same time, ultimately allowing the whole myofibril itself to shorten, which makes the muscle cell shorten. So if we can zoom in on this myofibril, remember, this is actually the protein cylinder that would fill your muscle cell. And each muscle cell or fiber contains thousands of these myofibrils. And we're just looking at a piece of the myofibril. Now, the sarcomere is from Z disc here to the Z disc on this side. So the distance between these Z discs is actually the sarcomere. And the sarcomeres are basically made of thick filaments, shown in red, as well as thin filaments that are shown in blue. And these thick and thin filaments actually overlap, and they interact with each other in a way that allows for this whole sarcomere to kind of slide and pull against itself, so that the Z discs get pulled closer towards the M line here, so that the whole sarcomere shortens. But because these are made of repeating segments of sarcomeres, the whole myofibril also shortens. So the I band is actually found here between two different sarcomeres. It's basically the space between the thick filaments on this side versus the thick filaments on this side. And the A band is basically just the distance of one thick filament from end to end. The H zone is basically the space here in the middle of the sarcomere, where the thin filaments don't quite touch, and there's a space between the thin filaments at either side. And the reason why we define these zones is that we can look at these in terms of how the sarcomere changes before and after contraction. And this is not in a contracted state. This is actually more of in a relaxed state. Now the myofilaments, we have actin and myosin myofilaments. The actin myofilament is also called the thin filament. That was shown in blue in that picture. The myosin myofilament is the thick filament. That's the one that was shown in red in that picture. Now the actin myofilaments extend across the I band as well as part of the A band. And these are actually anchored to the Z discs so that the actin myofilaments are the things that are being pulled on, which allows these Z discs to get pulled closer together within the sarcomere. And the myosin filaments are the thick ones, which extend the length of the A band, and they're connected at the M line, which is the very center of the sarcomere. So what happens is the myosin myofilaments have little elements that actually pull on actin, and that allows the whole sarcomere to shorten. Now, sarcomere in cross-section is more of a hexagonal arrangement of one thick filament surrounded by many thin filaments, like up to 6 of these. And we're just looking at one angle here of the actual sarcomere. Where in cross-section, you'd see that that's actually a little bit more complex and densely packed. So here's our thick filament, also called the myosin filament. And here's our thin filament, also called the actin filament. Now the myosin filament has all these little extensions here, which are called the myosin heads. And these are basically enzymes that use the chemical energy in ATP to produce a ratcheting kind of motion to grab on the thin filament here. And it basically pulls the thin filament closer towards the M line. Now, the myosin filaments have heavy chains that intertwine to form a tail. That's just the main tube of this protein chain. And the light chains form this myosin globular head group. Now, the head group is actually what uses the chemical energy in ATP to form what's called a cross bridge. And these cross bridges are basically when the myosin head forms a bond or association with the actin or thin filament. Now, myosins are offset from each other, results in a staggered array at different points along the filament. Now, the thin filaments are composed of a fibrous protein called actin. Actin is a polypeptide that's made of kidney-shaped G-actin globular subunits, and these actually have the active sites for the myosin heads to bind. And G-actin subunits link together to form long fibrous F-actin or filamentous actin, and 2 F-actin strands twist together to form the entire thin filament. So in reality, we're saying that the F-actin is made of, lots of G-actin that are all stuck together like beads in a chain, where if the G-actin is the bead, the F-actin would be the chain, and 2 chains or F-actin filamentous strands wrap around each other to form the whole thin filament. Now, we also find other proteins associated here like tropomyosin and troponin, and these are regulatory proteins that are bound to actin, and they play a role with preventing or allowing muscle contraction to occur when necessary. So if you look at the thick filament here, we see that we have our myosin heads. And our myosin heads are the light chain or globular portion of the myosin protein filament. And these are the ones that actually use the chemical energy in ATP to move back and forth. And they produce this ratcheting motion that allows them to grab onto a nearby thin filament and basically pull like little hands. So imagine these are all like little hands that are pulling on our thin filament. Now they're staggered in their position, which allows many different positions of myosin heads to pull on the 6 different thin filaments that would surround this one, thick filament. So on the thin filament here, we have our actin filament, and essentially, remember, it's made of 2 coiled F-actin strands, and these F-actin or filamentous actin strands are made of G-actin subunits. That's what all these are. Now, G-actin has the active binding site here for the myosin to attach to. So these little divots here are places where the myosin heads could attach or form an associated bond between the thick and thin filaments. In fact, if you go back here, you can see that your myosin heads, could fit pretty well within these active sites. However, if you look here, there's actually proteins that are blocking the active sites on this thin filament. So we have this long kind of gold-colored or yellow-colored protein here, which is basically called tropomyosin. Now, the function of tropomyosin is actually to block the active site on actin during muscular rest. That way your muscles aren't always contracted. And then troponin is a nearby protein that's associated with tropomyosin, and troponin's calcium sensitive. Now the function of troponin is to bind calcium, which causes a change in the conformational state of this protein that allows for tropomyosin to move out of the way to expose the active sites here. That way the myosin heads could potentially bind. But if the muscle's relaxed and there's not calcium present, troponin and tropomyosin relax and they basically block the active sites. So looking at an electron micrograph picture here, you can see this is the thick filament with all your myosin heads sticking off, and then here's our thin filament, which is what they're attached to. And basically you're looking at muscle in its cross bridge, where these two are connected to each other in their contracted state under an electron micrograph. Now the molecular composition of the myofilaments also include things like elastic filaments or dystrophin. The elastic filament here is a protein called titin. And titin is what gives muscle tissue its elasticity because it holds the thick filaments in place and it helps to recoil after stretch. Now titin also resists excessive stretching, so it plays a very important role in stability of your sarcomeres. Now dystrophin links the thin filament proteins to the sarcolemma. Remember, sarcolemma is the muscle cell plasma membrane. And if dystrophin is mutated or non-functional, as you find like in muscular dystrophy, we see that the muscle cell plasma membrane or sarcolemma is a little weaker and it's more likely that these cells can rupture and die. Now other proteins like nebulin, myomycin, and C proteins bind filaments and sarcomeres together, and they maintain proper alignment of the sarcomere. So they're very important proteins here. Now the sarcoplasmic reticulum is also another specialized feature of skeletal muscle cells. And you find these in a little bit in smooth and cardiac muscle as well. And it's basically a network of smooth endoplasmic reticulum tubules that surround each myofibril. Now most of these run longitudinally, and they have these terminal cisterns that form perpendicular cross channels at this AI band junction. And the whole purpose of this, you guys, for SR, is to store calcium. So sarcoplasmic reticulum serves as a storage site of calcium within muscle cells. And it releases calcium when necessary, and calcium is a stimulus for muscles to contract. The T-tubules are also highly associated with sarcoplasmic reticulum, and essentially what these are, and as they sound like, are a tube, which is a protrusion of sarcolemma, or membrane, deep into the cell interior. And what they do is they increase the muscle fiber surface area. Now they're continuous with the extracellular space, and they allow for electrical nerve transmissions to reach deep into the interior of each muscle fiber. These T-tubules penetrate the cell's interior at this AI band junction, and they form what's called a triad, which is basically the place where T-tubules meet the terminal cisterna of sarcoplasmic reticulum. Now this is significant because it's actually the electrical impulses or currents that go down the T-tubules, eventually getting to the terminal cisternae of your sarcoplasmic reticulum to trigger the release of calcium into the muscle cell, which allows for these muscle cells to begin a contraction. Now the triad here is important because these contain integral membrane proteins that protrude into the inner membrane space, and your SR is nearby, which allows for these integral proteins to control the opening of calcium channels in the SR, so that once electrical currents travel deep into these T-tubules, it can trigger the opening of these SR cisterns. That way calcium can leak out of the sarcoplasmic reticulum into the sarcoplasm and then bathe the myofibril in calcium. Now, this is actually all stimulated by an electrical impulse that travels down that T-tubule and thus causes a change in the shape of these SR proteins, which cause calcium to rush into the cytoplasm. So looking at a picture here, in blue is the sarcoplasmic reticulum, and these things that are kind of grayish in appearance are the T-tubules. You see that T-tubules dive down deep into the muscle cell, and they're associated with your longitudinal SR. Now what's important to note then is when electrical impulses or currents traveled through these tubules, all along the way they're triggering the sarcoplasmic reticulum to release calcium, and then calcium is basically going to get washed into this myofibril protein tube, which allows for its association with troponin to get the muscle to contract. So contraction is the activation of cross bridges to generate force. And shortening occurs when tension is generated by these cross bridges on thin filaments. And the contraction ends when these cross bridges become inactive. Now, we call this the sliding filament model of contraction, and it states that during contraction, thin filaments slide past the thick filaments, causing actin and myosin to overlap more. Now, neither the thick nor thin filaments change in length, but they just overlap more due to a change in their position. Now, when the nervous system stimulates the muscle fibers, these myosin heads are allowed to bind to actin, which forms a cross bridge, and it causes a sliding motion or contraction due to the ratcheting movement of these myosin heads. Now, cross-bridge attachments form and break several times, and each time they pull the thin filaments a little closer towards the center of the sarcomere in a ratcheting action. Now, this causes shortening of the whole muscle fiber, which then would ultimately cause shortening of the whole muscle. And these Z-discs are pulled closer towards the M line. Remember, Z-discs are on the side, and they actually are pulled towards the center of the sarcomere. The I bands will also shorten, and the H zones disappear when the thin filaments overlap. A bands also move closer to each other. So as we're seeing here, the sarcomere, from Z-disc to Z-disc, here's our A band, which is the thick filament with our myosin heads. When myosin heads bind to actin, they have a ratcheting motion which pulls on these thin filaments towards the M line, or the center of the sarcomere, and it's actually going to pull our Z discs closer together, causing this whole sarcomere to shorten up. Now remember, what holds your thick filament in place, the myosin filament in place, is this large elastic protein called titin, and it serves as a large molecular spring. Now, this is actually showing the sarcomere in a contracted state where the myosin heads have pulled on thin filaments towards the M line. Our H zone has disappeared because the thin filaments have overlapped, and we can see that our Z discs are now closer together, which means that the entire sarcomere is a little shorter than it was before.